Today we're going to be talking about probably one of the most important topics that we're going to cover in our study on evolution, and that is the idea of embryology, homology, and then we're going to look at the very end of the lesson on Haeckel's embryos. Why is this so important? Because Darwin considered this probably the most important evidence he had for evolution. In fact, I would go so far as to say that the theory of evolution fails or survives on the validity of the notion of common ancestry that was proposed by Darwin in his book on the origin of species. So common ancestry obviously is key in the belief of evolution. You know, starts molecules to man, and then everything there descended from that. So it's very important. But Darwin also knew that he didn't have the evidence in the fossil record. He knew that the fossil record was really not there to prove this concept of common descent. So where did he look for it? He looked for it in the idea of homology, the concept of homology. So for Darwin, this was key. It was probably the most important evidence that he had at that time for homology. Now, we're going to be studying in our next lesson the idea of homology through the DNA record, okay? And that's where evolutionists now are looking. Now, the evolutionists haven't given up on homology of structure. They haven't given up on that, but they understand that there is another explanation for that, which we will look at besides common descent and evolution. But now they're looking more at the DNA record, and we'll spend some time on that. Well, where did homology come from? It didn't come from Darwin. Homology had already been proposed by Richard Owen, who was a biologist in the 19th century. And he looked at the characteristics that animals had, the similarities that animals had, and he actually coined the word homology. He actually looked at another concept of anology. Anology is common function but not common structure. An example of that would be like the wings of an insect versus the wings of a bat. They do the same function, but they certainly don't look a lot alike, and they're structured quite differently. And other biologists held to this concept too. It wasn't just Richard Owen, but he's the one that came up with the words, but other prominent zoologists and biologists of the time, such as Louis Agassiz, held on to the same idea. Now, what was their concept of homology and their reason for homology? They looked at it as a common designer, if you will, okay? Not common ancestor, but a common archetype, or common designer. So they were not evolutionists. They didn't believe in evolution, but Darwin came along and sort of adulterated this idea of homology. And for Darwin, the best explanation for homology was a common ancestor. That's where he got the idea. Well, what is homology? Homology is similarity in structure. That's what we're talking about. Where animals, especially when you get into the mammalian class, are similarly constructed in various ways. That's what homology is. Anology, as I mentioned before, was similarity in function. Evolutionists have a harder time with that, because that doesn't necessarily make a lot of sense. If you take the concept of homology and common descent and things looking similar, that makes a lot more sense. But how you get an analogous structure is a little bit harder to explain for the evolutionists, and really they don't even try. But that's what homology is. So homology was absolutely apparent to Darwin. Similar types were symmetrical with, two eyes, two forelimbs, two hind limbs, shared similar organs and similar systems. And this was just obvious for Darwin in his belief on evolution. And I want to read a couple of excerpts from Origins to show how important this was to Darwin. He says, "We have seen that the members of the same class, independently of their habits of life, resemble each other in the general plan of their organization. Is it not powerfully suggestive of true relationship of inheritance from a common ancestor? And what can be more curious than that the hand of a man formed for grasping, that the mole for digging, the leg of the horse, the paddle of a porpoise, and the wing of a bat should all be constructed on the same pattern and should include similar bones in the same relative positions." So it was powerful evidence for Darwin, this similarity of pattern. And we'll look at this picture again in a minute, but this is the pattern that he mentions in the book, Origin of the Species, on the origin of the species. He looked at the bones of a bat and seal and other animals, and he looked that they all had sort of a 5-bone structure. And we'll talk about that in a minute. To me, it's not quite all that obvious, but if you look that we all got humeruses and radius and ulnars and things like that, and these animals have similar structures, and that similarity in structure would be called the pentadactyl design. And Darwin considered it kind of hopeless for creationists. He didn't see how creationists could explain this. It was a hopeless cause. He says nothing can be more hopeless than the attempt to explain this similarity of pattern in members of the same class by utility or by doctrine of final cause. The idea of final cause would be a creator. And he thought it was a hopeless cause to do that. And the reason he felt this way is, why would the creator, if there was a creator, be limited to this pattern? Couldn't he have used other patterns? Could you have animals with 3 eyes or 4 eyes, or mammals with 5 legs? Why would he be limited to that? And so again, for Darwin, this was a problem for creationists, a big problem. We don't have a problem with it, do we? But he did. So the fact that animals are homologous and show homologous structures is not the issue. Would anybody disagree with that? That animals share similar structures of homology, it's sort of evident. For creationist, homology is not the question. The question really is the cause. Why are we similar? I'm going to show a little picture here to show the difference now. This happens to be 3 different species. One is Homo sapien, me, and the other one is my dog, Bobby, and the other one is Cherry's cat, kind of my cat too, I guess, Callie. There's some similarity there, isn't there? Even though we're quite different. All these animals have 2 eyes, okay? We have 4 arms and hind limbs. And there's similarities that you can't see. We all have hearts. There's a heart beating in all 3 of those species. There's respiratory tract. There's a digestive tract, reproductive tract, so on and so forth. And so there are similarities in structure and in homology. That's really not the question. The question is, more importantly, is common ancestry the only or even the best explanation for homology? Homology is a mainstay in the textbooks of biology and evolution, or in high schools and colleges. So it is still a major concept. It is in all the textbooks. They will continue to put those in the textbooks. So the validity of an evolutionary explanation for homology would be strengthened if embryonic development were specified by homologous genes. Now, what does that mean? That's sort of a scientific thing. Well, that basically means that if you have the same genes in the animals producing the same structures in the animals, and they shared those common genes, that would at least give you a little bit more validity for your theory. It still wouldn't be enough, in my opinion, but it would give you a little bit of validity for your theory. But there's a problem, and the problem is, again, this would at least suggest, according to Michael Denton, a true relationship. And it is becoming increasingly clear that the principle cannot be extended this way. So it would at least again suggest a true relationship, but as Michael Denton says, it doesn't really seem that is proven. And why is that? First of all, homologous structures are frequently specified by non-homologous genes, and the concept of homology seldom extends back to embryonic development. What does that mean? That means the same gene, we may share similar structure, but the genes that produce those structures are different frequently. Not always, but frequently. So homologous genes don't always share homologous structures don't always share homologous genes. That's a problem then. That doesn't fit. That doesn't fit this embryonic development that we're talking about. Second thing, sometimes we see the opposite. We see non-homologous genes produce the same structures. So you got the opposite taking place. And then finally also, and I didn't mention this, but homology would also be strengthened if species shared the same germ cell formation. And we don't see that. The mode of germ cell formation varies frequently from one animal species to the next. So there's some problems with homology as far as looking at it from an embryologic standpoint and a developmental standpoint. That doesn't deter the evolutionists though. And they employ what we see a lot, and that's circular reasoning. Look at what Ernst Mayr said after, this is a quote from him, after 1859, there has been only one definition of homologous that makes biologic sense. Attributes of two organisms are homologous when they are derived from an equivalent characteristic of a common ancestor. Anybody see a problem with that definition? Well, it's a circular definition. They're homologous when they come from a common ancestor. Well, you haven't proved they come from a common ancestor yet. So it employs circular reasoning. And biologists, by the way, have known that these non-homologous genes produce homologous structures and vice versa. They have known that for some time. So they employ what I would call, again, classical circular reasoning. Circular reasoning works because circular reasoning works because circular reasoning works. And that is classic when it comes to the concept of homology. But I want to take one more look at this idea of the four arms of the vertebra. And again, this is in all textbooks, this pentadactyl structure. Again, on the far left, or my far left in the slide, would be a picture of a human arm. Once again, you have a humerus, a radius, an ulnar, then you have the carpal bones, and then they really throw the metacarpals and the phalanges into one bone. Even though the metacarpals and the phalanges are different, those are kind of in one system. And that's how they get the concept of five, pentadactyl. Then you have a cat. The cat has a humerus, cat has a radius and an ulnar, cat has carpal bones, and cat has phalanges and metacarpal bones. You've got the whale and the bat. It gets a little harder to see when you get into a whale, doesn't it? Those bones look a lot different, but still there is some commonality there with what they would call a humerus and a radius and an ulnar, and again, carpal bones and then metacarpal bones and phalanges, and then on the far right would be the bat with the same pentadactyl structure. So for Darwin, this basically was proof of homology and then proof of a common ancestor. Again, circular reasoning. You prove homology, you prove a common ancestor. Well, no, you don't. That's a circular reasoning pattern. Jonathan Wells calls this putting the cart before the horse. Okay, it doesn't prove anything when you just say homology equals common ancestor. All you've proved is that there's a similarity in structure. Alan Boyden in 1947 says that we first know the ancestry and then decide the corresponding organs and parts are homologous. That's what they do. And so again, a common thing that is done by the evolutionary scientist is assume one thing and without actually proving it and then use it to prove the other side. So their similarity does not always make evolutionary sense. I threw up there a picture, maybe a little bit hard to see, but believe it or not, the eye of an octopus and the eye of a human being look alike. There's a lot of similarity there, a lot of similarity. They're very, very similar. And this picture sort of depicts that. Again, it's a little bit fine to read, but on the left you have the human eye, and then on the right you have the eye of the octopus. The problem with that is no evolutionary biologist believes that the octopus, which is obviously not a mammal, and human beings come from the common ancestry, come from the same line. So how would you explain that? How do they get an eye that looks a lot alike when they don't come from the same line of development? And philosopher Ronald Brady points out that by making our explanation into the definition of a condition to be explained, we express not scientific hypothesis but belief. We are so convinced that our explanation is true that we no longer see any need to distinguish it from the situation we are trying to explain. Again, the concept of circular reasoning. Some people have used the fossil record to try to show this common ancestry. And they go back to the fossil record. It's really, without the significant transitional forms, it's impossible to show that. What bugs me a little bit is that I hear, and we saw in our film 2 weeks ago, they kept asking about transitional forms, right? And how many transitional forms did they show that were not anything more than microevolution? I see that all the time, though. I was reading for our next study. We're going to be looking at DNA. I was rereading Francis Collins' book on the language of God. At least 3 times in there, he had pointed out that transitional forms have been found. He never gave an example of that. You know, to say it doesn't make it true. Just to keep saying, well, they've found transitional forms. In fact, they should have found millions of transitional forms by now. So even if they allude to 2 or 3 that they think may be transitional forms, that doesn't prove there's transitional forms. You know, there should be millions of transitional forms. And when you look at the difference in DNA from species to species, this should be quite obvious. But a fellow came along by the name of Tim Berra, B-E-R-R-A, Berra, and this has been referred to as Berra's blunder. In 1990, he published a paper to try to show what is meant by evolution and the idea of transitional forms and what the fossil record should show and what the fossil record does show. And this is some pictures of corvettes. And everybody knows what a corvette is. You know, it's been around since the 1950s. And I think the bottom picture there shows the earlier versions of the corvettes. And you got the earlier versions, and you've got them changing into, well, through various stages throughout the years, because the corvette is still around, isn't it? And so you have the modern corvette at the very top. And what Berra was trying to show is this is how evolution works. It goes through these stages, okay, where you go from one stage to the next stage to the next stage to the next stage. Well, what's the problem with that picture? Do what? Right. This doesn't prove evolution. This actually it helps the creationist, because we know that the corvette did not evolve. It didn't start off with this one and slowly change in the other one over time. Man changed the design. So it became known, Phil Johnson referred to it as Berra's blunder, because it actually, there was just as good an explanation from a creationist standpoint as an evolutionist standpoint to show this concept. So you'll see that sometimes. That explanation has been around a long time. Berra just put it in print. So homologous structures produced by similar developmental pathways in the embryonic state just does not fit the evidence. So what the evolutionists say is that when we look at the germ cell development and look at each species as it develops from an embryonic state, that we can see evolution in that process, but that really doesn't fit the facts. Why? Because from the very, very beginning of fertilization, organisms look quite different from what we call the blastula stage, which is after the creation of several different cells. Even within the same order, they don't look the same. They look quite different. That doesn't fit from an evolutionary standpoint, does it? There is great dissimilarity in the early embryogenesis and the different vertebrate classes, and homologous structures cannot be traced back to homologous cells. And then we talked about this again, but there's another problem. Almost every gene structure in the higher, well, we haven't talked about this is a different concept. But another problem pointed out by Michael Denton is almost every gene structure in higher organisms has been found to affect more than one organism system. And this is called pleiotropy. So what does that mean? Okay? That means the genes, you have different genes affecting the same structure. Again, I will reread that. Almost every gene structure in the higher organisms has been found to affect more than one organ system. And that doesn't make sense from an evolutionary standpoint, when you got genes affecting more than one organ system. And he points out this process is called pleiotropy, or the pleiotropic effect. And it is, as he points out as well, is that pleiotropic effect is species specific. So if you've got evolution taking place, how can you have these genes affecting more than one organ system? And if they come from the same gene, and those genes are different from species to species. Doesn't make sense. And then finally, also, there's another problem with this one we have already speaked about, and that's the idea of analogy or anology. What do we mean by that? Again, now we've got different structures that are clearly from different genes but doing the same function. So you've got, in these pictures, you've got a bird on our left, insect on our right, top right, a mammal or a bat on the bottom left, and then a flying fish. That's what that is on the far right lower column. What do these have in common? No, they're flying in some way or the other. They're flying. Their wings and their method of flight are very different, quite different. And so what you would have to have from an evolutionary standpoint is you would have to have flight, the ability to fly, evolving at least in 4 different ways in 4 separate lines. Now, the evolutionists said, well, it could happen, and they would even argue, well, it did happen, okay? But that would be a little bit difficult, more difficult to explain. Also, that pendactyl design has problems too. That pentadactyl design that we talked about has problems too. And I hadn't thought of this till I read, again, Michael Denton's book. And that is that the forelimbs of terrestrial mammals follow this design, but so do their hind limbs. Their hind limbs are structured similarly as well. So you're going to have, and from an evolutionary standpoint, both the forelimbs and the hind limbs developing similarly at the same time with the same design. Kind of stretches the imagination. Michael Denton says, no evolutionist claims that the hind limb evolved from the forelimb. They evolved separately, but with the same similar pattern. Or that the hind limbs and the forelimbs evolved from a common source. Do you see why that's a problem? You'd have to have both the hind limbs and forelimbs developing at the same time and looking similar, because no one believes that one came from the other. So how this complex and seemingly arbitrary pattern could evolve twice independently is, quote, mystifying. That comes from Michael Denton. So unity of type is just not readily explained by evolutionary theory. We don't deny that there is unity of type. We just deny that it's explained better by evolutionary theory. Well, what would explain it? What does the unity of type or homology point to if it's not evolution? How about a common designer? Wouldn't that explain it? Couldn't God have used the same pattern to produce animals, similarly? Yes, he could have, and he did. So I believe it's a common designer, not an evolutionary concept that explains homology. Why should it be surprising? He could use similar and homologous structures in his design of animals. We know, for example, again, we're going to speak on this next time we meet, but every life, plant life and human life for that matter, animal life, insect life, bacteria life, every animal life on the planet uses DNA. That is if you want to use the word language of God, it's the way God produced life through DNA. So if he used a similar chemical or molecule to produce life, why should it be surprising that there could be similarity in structure? I don't see that as a big surprise. The fact that DNA is around and the fact that we're all made of DNA and all the animals and plants are made of DNA is an absolute fact. So I believe that there is a common design and a common designer. That's homology in a nutshell. And there's no good biological evidence that evolution is the only explanation for homology. What separates man from the other animals? Well, it's not our flesh. We're all flesh. We all have DNA. All animals, and man included, have DNA. And again, we have similar structure to other animals. And some animals we look closer than other animals. For example, let's look at the other primates, the chimpanzee and the gorillas. We have a little bit more similarity to those than we do other animals. Wouldn't you agree with that? And you know what? When you look at the DNA structure, the DNA is more similar. Well, evolutionists jump on that and they say, well, see there, we're closer to the chimpanzees than we are the mouse or the whatever animal you want to pick. Why would that be surprising? If we look alike, wouldn't you expect the DNA to look similar? So what does separate us? Well, we know that it's the spirit. There is something different about human beings that's different from other species or animals, isn't there? God created man in his own image. In the image of God, he created him. Male and female, he created him. I don't believe that verse is talking about the physical structure of man looking like God. I just don't believe that's what that verse is talking about. We're made in the image of God in that we have a spirit and animals do not. And we have a moral code and we have other things that animals just do not have. So at best, homology gives only a circumstantial evidence for evolution, and circumstantial evidence can be quite deceiving. That is at its best, it's circumstantial. I love this quote again from Michael Denton's book, and he quotes Conan Doyle's Sherlock Holmes. Circumstantial evidence is a tricky thing, answered Holmes thoughtfully in the book. It may seem to point very straight to one thing, but if you shift your point of view a little, you may find it pointing to an equally uncompromising manner to a different, something different entirely, something entirely different. There is nothing more deceptive than an obvious fact. So we're going to spend the last 10 minutes of our class looking at Haeckel's embryos, because this is in the textbooks too. Haeckel or Haeckel, I'm not sure how to pronounce it. He was a German biologist. In fact, he's been called Darwin's German bulldog, kind of like Thomas Huxley was Darwin's bulldog. This is Haeckel's German bulldog, or Darwin's German bulldog, and he looked at embryos. And so Darwin said, "It seems to me the leading facts in embryology, which are second to none in importance, are explained on the principles of variation and the many descendants from some one ancient progenitor. Continuity of embryonic structure reveals continuity of descent." Again, that's sort of a technical term, a scientific term, but as the embryo develops, the embryos should look and act just like the embryos of other animals, have similarities there. And so again, ontogeny recapitulates phylogeny. It's kind of hard to say. Ontogeny recapitulates phylogeny. This came from Ernst Haeckel. You notice a lot of these Germans have the first name Ernst, kind of peculiar. Anyway, they have similar first names. But what does he mean by that? This was referred to, and these terms, ontogeny and phylogeny, were actually invented by Dr. Haeckel. And he referred to this as his biogenic law. And this is what that quote from Darwin that we just read is basically saying. And he's saying, on the way to birth, a human becomes a fish, an amphibian, and so on up the evolutionary ladder. This is a quote from Ernst Haeckel. So ontogeny, what's the concept of ontogeny? Ontogeny designates embryonic development, and phylogeny designates the evolutionary history. So to put it in other words, as a species develops, it goes through all the evolutionary processes that have gone on hundreds of million years before that. And so you have to go through those stages. That's what the biogenic law talks about, ontogeny recapitulating phylogeny. I had thought that this had been given up a long time ago. I thought biologists had given this up a long time ago. They haven't given this up. It's still in the textbooks. I think people that know better have given it up, but it's still in the textbooks. So what Ernst Haeckel did is he looked at the embryos, and these are not labeled, or they're too small for me to read, but these are various species. These are salamanders, these are other species, including on the very, very far right, you've got a human species. Somewhere in there, I think you have the canine species. And he looked at the embryos, and he stated that at the very earliest stages of development, these embryos look a lot alike. And I would suppose that you could say they do. If you look at the top row, wouldn't you say those look similarly alike? And yet one of them on the far left is a salamander, and the one on the far right is a human species. And you've got these 7 or 8 different species there in his original work. And he drew these species. He drew them out on paper. And he also made wood cuttings of them to show their similarity. Now when you get to the bottom, they look quite different. In fact, you can start to see a chicken in one of them, can't you? And again, the human on the far right, and these other animals, salamander and other animals here as well. And this was proof to him that, in fact, that ontogeny does recapitulate phylogeny. And also up there on the top of the board, you see what looks like sort of little gills. You know, these little arches. You see that? He called those gill slits, and he put those in. There was a problem with Haeckel's drawings. First, Haeckel purposely chose embryos that did look similar to depict only 5 of the 7 vertebrate classes for his drawings. He didn't include all the vertebrate classes, and the reason he didn't is because they would have looked quite different. So he wanted this to fit. It's kind of what scientists do when they're trying to prove a point. Probably more importantly, though, is he faked the drawings. Okay, that's probably more important. He fudged. Okay, he cheated. and it took a few years for people to understand that. So there's a problem when you look at these drawings in a textbook. You got to realize that the drawings originally drawn by Haeckel were not accurate. They were faked. But there's some other problems too. The fact that he faked the drawings is not really even the biggest problem. First of all, Haeckel labeled the first stage of embryonic development in his drawings, and it was no such thing. Those were not the first stages. Let me go back. These stages at the top are not the first stages of embryonic development. This is down the road from embryonic development. You understand? So those are not the first stages, even though they may look similar. And even though he fudged the paintings, you could still say there is some similarity there, okay? But the problem is that's not the first stages. At their earliest stage, need to go back to, at their earliest stages, embryos are very dissimilar, okay? And we've already talked about that. In the blastula stage, they are quite dissimilar. And this would, to me, this would cause one to reason more appropriately that they did not come from a common ancestor. If the blastula stage and the early stage of embryonic development looked a lot different, then this would give more credence to a creationist point of view, wouldn't it? And so there's a problem there. Now there is a time that embryos do have some similarity in appearance. And we talked, again, those pictures that we showed earlier, again, do have a similarity in appearance. And no one would deny that there can, you looked at those, there can be some similarity in appearance. And they do have that. But they don't have it at their earliest stages, which would go against evolution. Now, as we get further in development, you saw the bottom pictures where they did look quite different, then they start to diverge. This has been referred by Rudolf Raff as the developmental hourglass. I didn't have a real good picture of the blastula stage, but at the top of that hourglass is the blastula stage. And even according to the biologist, even a relatively untrained eye would be able to see that difference in the blastula stage. As they converge and progress, there is more similarity. I mean, not 100% similar and can still be distinguished quite easily, but from the naked eye would look similar. But then as you go along, then we become quite dissimilar, and it shows more of the adult stage, if you will. Point is, there is no evidence of organisms recapitulating their evolutionary history. The gills that Haeckel painted into the embryos are actually brachial arches that do occur in all animals, or most mammals at least. And these brachial arches are clefts or pouches. They've been referred to as the same thing. Once proposed as gill slits, don't ever become gills, even in fish. They're not even gills even in fish. Problem that I see, and you go to your standard textbooks, you will still see this stuff. It's still being labeled, and you still see Haeckel's embryos. And they still continue to refer to ontology recapitulating phylogeny. And to show my point here, this comes straight from a biology textbook by Peter Raven and George Johnson in 1999, fairly recent. Notice that the earliest embryonic stages of the vertebrates bear a striking resemblance to each other. Some of the strongest anatomical evidence supporting evolution comes from comparisons of how organisms develop. In many cases, the evolutionary history of the organism can be seen to unfold during its development, with the embryo exhibiting characteristics of its ancestors. That is just not true, but it is in every, or I don't know about every biology textbook, I haven't read every one, but in most biology textbooks, this is still being used. And a lot of the biologists, and some of the highly trained biologists, and some of the most highly educated biologists don't even know some of this stuff that we talked about today. A fellow by the name of Douglas Fudiyama, who is a professor of evolutionary biology, in a graduate textbook, pointed out the gill slits in the embryos and alluded to Haeckel's embryos. He didn't know that Haeckel had fudged those pictures until a creationist told him about it. So this stuff is still being used as evidence for evolution when it is no such thing. It's evidence, in my opinion, one more time, of a common designer. So finally, homology, embryology, and Haeckel's embryos continue to be used in college and high school textbooks as evidence for evolution, but as we have seen, only through circular reasoning can that fly. And so when somebody tries to convince you that evolution is true because of the similarity of structures that we all have, the answer should be yes, I know that. We do have similarity in structure. That points to a common designer, not evolution. So next time we meet, we're going to be getting into what may be a little bit of a deeper subject. We're going to be looking at DNA, because evolutionists now just believe DNA slams the door as far as whether or not evolution is true or not. They say the DNA records and looking at the chromosomes, even if you don't buy homology, even if you don't buy the fossil record as being evidence, DNA absolutely proves it. And we're going to look at that in some detail the next time we meet. So thank you for your attention.